Round 2 with notes. I'm going to go a whole lot slower. We're still going to talk about cell organelles, so get out your notes. And this time, I hope you take them because I want you to study with them later. Ready? Let's do it. The first is a cell membrane, or also known as a plasma membrane. It controls what goes in and out of the cell. It is sometimes called selectively permeable because it controls what goes in and what goes out of the cell. It selects. The structure is made up of phospholipids in a bilayer, which means there's 2 layers. There's phospholipids on top and there's phospholipids on the bottom. We'll talk more about cell membranes later. Cytoplasm. It is a jelly-like fluid or gelatinous type fluid that holds the cell organelles in place. It's found on the inside or interior of the cell. It contains dissolved solutes needed by the cell. The fluid that suspends the organelle is the cytoplasm. The structure of the cytoplasm is a watery, jelly-like substance. The nucleus. Its function is to protect the eukaryotic cell's DNA. DNA is the blueprint for cells' ribosomes. When the cell needs to make proteins, a copy of the instructions are sent to the ribosomes. The structure of a nucleus is large, round, circular structure enclosing the cell's DNA. It's found both in plant and animal cells. Ribosome's main function is to receive copies of instructions from the cell's nucleus. It bonds amino acids together to build a cell's protein. Attached ribosomes send newly assembled proteins to the rough endoplasmic reticulum, or rough ER for short. The structure is scattered in the cytoplasm and cover the surface of the rough ER. Ribosomes are small, but most numerous organelle. Most numerous in cells with high protein demands like muscle cells or islet cells, spelled I-S-L-E-T, of the pancreas, that make and secrete insulin, a protein, and digestive enzymes, which are also proteins. Golgi apparatus, also known as a Golgi body or simply a Golgi. They receive proteins from the rough endoplasmic reticulum and lipids from the smooth endoplasmic reticulum. When they receive them, they package them, sort them, and ship them out to their final place in the cell. Some proteins and lipids become phospholipids. Some proteins and lipids become embedded in the cell membrane. And some proteins and lipids become or form a barrier. Lysosomes. I like to think of them as lysol because of what they do. That helps me remember their function. Anyway, lysosomes, their main function is to destroy unneeded material like old cell parts and engulf viruses and bacteria. Their structure is vesicle wrapped in a membrane. Enzymes were made and modified by ribosomes in the rough endoplasmic reticulum, formed from the part of the Golgi apparatus. They are most abundant in cells that engulf foreign material like white blood cells. Smooth endoplasmic reticulum, or smooth ER. Its function is to assemble lipids for the body, like cholesterol and phospholipids. Sends lipids to the Golgi to be modified and tagged. Its function is to detoxify drugs and poisons from the body. It's most abundant in the liver, where the blood is filtered. The structure is smooth tubes with no ribosomes on them. Chloroplasts, found in plant cells, not animal cells. Its function is to perform the process called photosynthesis. Photosynthesis uses light energy to synthesize glucose, or food, for plants. It uses water plus carbon dioxide plus sunlight to make oxygen and glucose. It is interdependent with the mitochondria. What one makes, the other one takes. The structure are large, green, circular stacks that contain thylakoids. Mighty mitochondria. Its function is to perform a process called cellular respiration. Respiration makes a usable form of energy called ATP, or adenosine triphosphate. It uses oxygen plus glucose to make water, carbon dioxide, and ATP. It's found in all eukaryotic cells, but it's most numerous in cells with high energy needs, like muscle cells. It looks like a kidney bean. Cytoskeleton. Its function is to provide support that vesicles move along. It provides structure to the cell used during mitosis to move and separate parts out of the cell. Proteins and lipids are shipped to the vesicles that are moved along the cytoskeleton. The structure are thin filaments that crisscross all over the cell. Centrioles. Their function is to organize the DNA when the cell needs to divide. It assists in cell division. Its structure are two short cylinder-shaped structures that move on the opposite sides of the cell when it is ready to divide, like during mitosis and meiosis. Cilia functions to beat to move the cell in different locations. It also helps obtain dust particles. Cilia found on cells along airway and lungs to move dirt and debris upward. Their structure are short, hair-like structures that extend from the surface of the cell. Its function is to whip to move the cell. The structure are long, thin, tail-like tail. Cell wall. Bacteria and plants have a cell wall outside the cell membrane. Its function is to add structure and support to plant and bacteria cells. The structure is a tough layer outside the membrane in plant cells. No cell wall is in animal cells. Vacuole. Its function stores water, food, waste, and other molecules. The structure, plant cells usually have one very large storage vacuole. Animal cells can have many small storage vacuoles.